Good morning, student. Today we are going to discuss translation inhibitors and few points on post-translation modification. Now, we have already discussed prokaryotic translation, different mechanism like initiation, elongation, termination, as well as eukaryotic initiation mechanism. As elongation and termination for both prokaryotics and eukaryotics are similar, so we have discussed only one mechanism for that. Now, translation inhibitors are, if this is the translation mechanism which is happening inside a ribosome, and this is the mRNA, you can see that P site tRNA is there, at A site also tRNA is there. So, different ribosomal subunit or different mechanism during this entire translation process can be a target of different inhibitors who can inhibit translation at different stages. So, as a protein synthesis inhibitors are, most of them are antibiotics. Antibiotics are bacterially, fungally, or sometimes synthetically produce substance that inhibits the growth of microorganism. Antibiotics are known to inhibit a variety of essential biological process including DNA repair, transcription at different stages. However, the majority of known antibiotics, including a great variety of medically useful substances, block translation. The gist of the thing is that most of the antibiotics which are present, which we are seeing every day in our life, are their target is our translation machinery. Now, why the translation machinery? Because the translation machinery are very vast and there are variety of factors are involved, so they could be the target of such antibiotics. This situation is presumably consequence of the translational machinery's enormous complexity, which makes it vulnerable to disruption of in many ways. That means there are so many complicated mechanisms are there, so many proteins, so many factors are involved, so antibiotics can interrupt any that the chances of getting its target more and more. Here are some list of some inhibitors or the antibiotics, and whether they are prokaryotic and eukaryotic, you need to remember. Inhibitor chloramphenicol is an inhibits peptidyl transferase on the prokaryotic target large subunit. Cyclohexamide is an eukaryotic translation inhibitor. It inhibits peptidyl transferase on the eukaryotic large subunit. Like that, erythromycin inhibits translocation by prokaryotic large subunit. Then fusidic acid inhibits elongation in prokaryotes by binding to EFG GDP in a way that prevents its dissociation from large subunit. Then come paromomycin, increase the ribosomal error rate. Puromycin, an aminoacyl tRNA analog, it mimics an aminoacyl tRNA that causes premature chain termination in prokaryotes and eukaryotes. So, puromycin is the target for both eukaryotes and prokaryotic translation inhibitors. Streptomycin causes mRNA misreading and inhibits chain initiation in prokaryotes, as well as tetracycline also inhibits binding of aminoacyl tRNA to the prokaryotic small subunit. The next one is diphtheria toxin, catalytically inactivates. This is eukaryotic elongation factor 2 by ADP ribosylation. We will study. Then come ricin and abrine are poisonous plant glycosides that catalytically inactivate the eukaryotic large subunit by hydrolytically depurating a specific highly conserved A residue of the 28S rRNA, which is located on so-called sarcin rich ricin loop that forms a critical part of the ribosomal factor binding center. Now we will study few of them in details. First we will see the function of puromycin, which is an inhibitor of both eukaryotic as well as prokaryotic translation. Now here goes the mechanism of puromycin, how it inhibit translation for both prokaryotic and eukaryotic. Puromycin terminates translation by mimicking a tRNA at A site. Now you can see this is a ribosome where at the P site tRNA is there and the growing polypeptide chain is there. Now puromycin can act as a tRNA and can enter through A site. Once it enters through the A site, there is a peptidyl bond synthesis also can takes place. That is also that is we named as peptidyl puromycin. So because of this formation, instead of the correct tRNA to come and bind at the A site, it competes with the amino acid tRNA, and being a small molecule, it enters from the A site, and once it occupies A site, it catalyzes. I mean, the whole system recognize it as a mistake, and it transfer the growing polypeptide chain to the peptidal and form a peptidal puromycin. Because of that, it's a small molecule, it diffuse out from the ribosome and a premature chain termination reaction takes place. So please remember, this is one of the very important thing you need to remember. There is one more thing we need to discuss, that is diphtheria toxin. So diphtheria toxin, it's catalytically inactivates elongation factor, eukaryotic elongation factor by ADP ribosylation. Remember, so let us see the mechanism. What is diphtheria toxin? We have studied in your third semester cholera toxin as well as pertussis toxin and their mode of function, how they actually regulates different cell signaling mechanism. Now diphtheria toxin, it is a disease, it's an respiratory tract infection disease resulting from bacterial infection, Corynebacterium diphtheria, that harbor that bacteria actually contains a bacteriophage. The phage is Corynebacterium beta. Diphtheria was a leading cause of childhood death, and although the bacterial infection is usually confined to the upper respiratory tract, what happened? The bacteria secrete a phage encoded protein. So remember, the protein which is named as diphtheria toxin, it's a phage encoded protein which is responsible for the disease lethal effect. Now diphtheria toxin specifically inactivates the eukaryotic elongation factor. Which one? Small e stand for eukaryotic elongation factor 2, thereby inhibiting eukaryotic protein synthesis. So this is the mode of action of diphtheria toxin. Now little details about its protein, but what we are expecting from you to write the mechanism, how actually it's inactivate the eukaryotic elongation factor. Now little bit mode of action, how it enters. It's actually a monomeric protein, 535 residues proteins. Once it's all ready to infect or enter the cell, it actually divide. A tryptic cleavage takes place, and it's become an A subunit and B subunit. A subunit and B subunits are actually linked with a disulfide bond. Now B subunit fragments has a receptor, okay, the A and B, which are nevertheless remained linked by a disulfide bond. The B fragment C-terminal domain binds to a specific receptor on the plasma membrane susceptible cell, thereby inducing diphtheria toxin uptake into the endosome. So a receptor mediated endocytosis takes place, and you remember diphtheria toxin is a phage protein. It enters inside the host cell. Once it is entered, because of the low pH, now the disulfide bond gets cleaved. So B and A, both the subunits are there, but the culprit is A fragment. Now, once entered in the cytoplasm of the host cell, the A fragment into cytoplasm, what it does, the disulfide bond linking the A and B subunit is then cleaved, and then A fragment starts the A fragment starts a catalytic behavior, catalyzes ADP ribosylation of whom ADP ribosylation of elongation factor, eukaryotic elongation factor 2. So see, and that ADP ribosylation, ADP ribosyl group, it is taking from NAD plus. So what is happening? Diphtheria toxin, it's in presence of NAD plus, it's take the ADP ribosyl and put it in the histidine residue of a particular histidine residue of the EE2 EF2. Now this is the reaction, thereby inactivating the elongation factor. Since the A fragment acts catalytically, one molecule is sufficient to ADP ribosylate all the cells E2F, which halts the protein synthesis and kill the cell. Now this is ADP ribosyl group, which actually coming and binding to the histidine residue, and this fragment is named as ADP ribosylated diphtamide. So this is the function of diphtheria toxin, and because of that it inhibit the protein synthesis. That's why it's impart its inhibitor effect. Now after this discussion, these two inhibitors, now we are moving to post-translational modification. Now post-translational modifications refer to any alteration in the amino acid sequence of the protein after its synthesis. Now the protein which is getting synthesized from the mRNA, that's not the exact protein which is actually functioning inside a cell. There are lots of modifications should take place to protein become a mature protein, a functional protein. So the modification range can be a vast. It may involve the modification of the amino acid side chain, terminal amino acid or carboxyl group by means of covalent or enzymatic means following protein biosynthesis. There may be removal of the N-terminal peptide. You must have studied pre-pro protein synthesize, and then how from the pre-pro it modified signal peptide hypothesis. So these are all post-translational modification. Glycosylation, hydroxylation, all these are modification. Then the different subunit also can join to become a functional protein. Multimeric protein also generate. Generally, these modification influence the structure, stability, activity, cellular localization, or substrate specificity of the protein. Post-translational modification provides complexity to proteome for diverse function with limited number of genes. So we have gene from gene to protein we are getting. Maybe after modification we may get diverse function from a single gene. Now here in this slide, what I'm showing, the ribosome mRNA, from that the protein is synthesized. Once the protein is synthesized, it should fold in a proper orientation, 3D structure, and then the modification takes place. At the simultaneously, hydroxylation attach a hydroxyl group to a side chain of the protein. Methylation takes place, and methylation may cause many alteration in the protein. Lipidation, such as lipids such as a fatty acid to a certain protein chain, can be also associated. Acetylation, we have discussed many things about how the protein gets functionally, it is activated or silenced or something based on the acetylation of the lysine residues. Disulfide bonds, you have one example you please try to give. Disulfide bond and stability covalently linked with sulfur atoms of two different cysteine residues. Then the simulation, recently we studied the SUMO protein, a small protein which may function like an ubiquitin, but actually it's not the alone function in degradation. Different other things also can take place after simulation. Ubiquitination adds ubiquitin to lysine residue of a target protein for degradation. Glycosylation attaches a sugar, usually an ene or oxygen, in an amino acid side chain. Phosphorylation can make a protein different amino acids, serine, threonine, or tyrosine, and then different function also it start behaving. Now generally the question come with a specific example, explain the post translation modification. You can take any name of a protein, you can take an example of insulin and can describe how insulin gets synthesized and being a secretory protein, how become disulfide bonds are forming, pre-pro insulin packaging, all these things you can discuss. You can discuss by taking another example like collagen. So collagen can be a classical example when we are discussing post translation modification. So there is synthesis of, actually it is a triple helix, so synthesis of single peptides, and then three peptides are coming and forming a triple helix, and the triple helix also cross link, and there is a cleavage at the N terminal side and the C terminal side. So entire process, then secretion of this protein, glycosylation, modifications, so many post translational modification can takes place throughout this journey of procollagen to its complete collagen protein and as a secretory protein. So we will discuss little bit in brief. Collagen synthesis is characterized by the presence of an unusually large number of co-translational, meaning at the same time of translation also some modification takes place, and post-translational modification, many of which are unique to collagen. You can divide in two parts. One is intracellular modification together with synthesis of the polypeptide chains result in the formation of triple helical procollagen molecule, remember, and then extracellular processing then convert these molecules into collagens and assemble the collagen molecules into a stable cross-linked fibrils. Now, the processing of non-fibrillar collagen is basically similar to that of a fibrillar collagen, except that it may not involve a procollagen intermediate, and extracellular supramolecular assemblies are not fibrils. These processing reactions require at least 9 specific enzymes and several non-specific ones. Let us see what those are. The intracellular modification requires 3 specific hydroxylase. What are the hydroxylase? Please remember these names. Hydroxylase that convert certain proline and lysine to 4-hydroxyproline. Proline can be converted to 3-hydroxyproline and then hydroxylysine. So 3 specifics are there, and 2 specific glycosyl transferase that converts some of the hydroxylysine residue to galactosyl hydroxylysine and some of the galactosyl hydroxylysine residue to glucosyl galactosyl hydroxylysine. So see how complex glycosylation takes place at lysine. The extracellular modification requires 2 specific procollagen proteinase. You must be remembering that. One to cleave the N-terminal propeptide and the other C-terminal propeptide. So that is synthesized as a pre-pro. And lysyl oxidase, which initiate cross-link formation by catalyzing oxidative deamination of certain lysine and hydroxylysine residues to reactive aldehyde derivatives. So how the intramolecular cross-linking takes place, that is mentioned here. One of these enzymes, procollagen N proteinase, has 2 collagen type specific isoenzymes, whereas in all other specific post-translational modification, a single enzymes appear to act on all the collagen types. Most of these 9 enzymes are now well characterized, and 3 of them have been cloned and the data are already available. Now, after these post-translation modification for particularly one particular example, I'm not going to tell you something different, that is a protein splicing, that is also we consider as a post-translational modification. You must have heard about RNA splicing. That means that from the DNA there is some non-coding part, that is we call intron, and then the exons are there. Intron excised off and exons are linked together, that is we call splicing. Like that only, for certain proteins, people have in the year of 1989, this is the time when this discoveries started coming, that not only RNA, the certain proteins also be spliced, okay, but not the mechanism is not exactly similar to what we know, that intron exon splicing. Now the DNA part of the protein named as extine, and the intronic, that means which is non essential, that part named as intine. So we are showing here the DNA from the DNA RNA and from the RNA protein. You can see there is a sky blue color precursor which we call intine, and the C-terminal, N-terminal black color, these are the extine. So after the protein splicing, this molecule excised intine, and the mature protein what you are getting, this is in this N-terminal extine and C-terminal extine get ligated, and we are getting a mature protein. The protein splicing were discovered for proteins in all three kingdoms of life. In eukaryotes, 69 kilodalton subunit of vascular ATPase of yeast. That's a classical example. In prokaryotes, REC-A proteins of Mycobacterium tuberculosis and Mycobacterium leprae. So some other examples also there. It's not so uncommon thing. So need to remember that protein splicing is also there. With this, I'm finishing today's lecture. Thank you very much.